Hi friends, this video is about nutrient uptake by plants. The focus will be on various passive modes of mineral absorption in plants. We'll discuss here different modes of passive absorption, that will be diffusion, facilitated diffusion, ion exchange, and Donnan equilibrium. First of all, let's understand what we mean by the absorption and uptake of mineral elements. This we have already understood, but we'll have to understand where are the mineral elements. As you are well versed that soil serves as the main reservoir of mineral salts, and where it can occur either in dissolved or combined state or in adsorbed state. So dissolved nutrients forms soil solution with soil water. So we have mineral elements here that can be in the form of a combined or dissolved form, or it can be in the form of an adsorbed state. So dissolved nutrients forms basically soil solution with soil water. Adsorbed nutrients form complexes with inorganic and organic substances like cations, potassium, calcium, magnesium, iron, etc. And they are held by negatively charged surface of colloidal clay. So we have the elements here which will remain with the colloidal clay, or they will be held with organic particles or humus, hydrated colloidal organic particles or humus by means of negatively charged ends. So we have the minerals in the soil. That's why we say soil serves as a reservoir of mineral salts. Either it can be in the form of a dissolved state, or it can form a adsorbed state. So mineral anions like nitrate, chloride, sulfate, bicarbonate remain in free state in the soil solution. So there are always chances of they'll be quickly leached out. We have a phosphorus which is tightly bound to surface of soil particles through adsorption process. The binding takes place on clay surfaces or iron and aluminum oxides and hydroxides present in soil. So you will see that adsorbed phosphorus, which is called as the sorbate phosphorus, can be released through desorption process into soil solution. It will be available for plant uptake then. So we have a calcium with sulfate that forms slightly soluble gypsum, calcium sulfate. So we have adsorbed mineral ions. They can be replaced by ions having higher concentration in the soil solution. Let's understand. See, we have given a passing reference about the phosphate. The phosphate source in the soil comes from the animal manure, plant residues, organic fertilizers. This is the phosphorus input, which goes into the soil as an organic phosphate, soil organic matter, microbes, soil biomass that can undergo the mineralization process. With the mineralization process, there will be the soil solution phosphate which will be available to the soil now, and it will be taken by the plants. That will be plant uptake, or some of it can be leached out, which will be a phosphorus output. Phosphates can remain adsorbed form. That's called the adsorption. It can be adsorbed with clay minerals, iron, and aluminum oxides. That is called as a sorbate phosphate. Or whenever needed, it can undergo a reverse process, which is called as the desorption process, and it will be taken by the soil again. In addition to this, the soil phosphate pool can undergo a precipitation and can remain in the form of a secondary phosphate minerals with calcium, iron, aluminum phosphates. Of course, there can be an interchange of this also, and it can again undergo dissolution and will be taken back to the soil pool. as a soil phosphate pool. And there can be also these phosphates can remain as the form of the deposits, which is called as the primary phosphate minerals, apatite, and that can also undergo, under certain circumstances, the process of weathering and will be released back to the phosphate pool, which is in the form of the soil solution. So phosphate can remain in an adsorbed form, or it can be in the form of a solution within the soil. Now we have the soils which can be acidic soils or which can be alkaline soils. So acidic soils can be made alkaline by adding lime to the soils, or alkaline soils can be made acidic by adding sulfur. When you add sulfur to the soil, the microbes, sulfur is oxidized to sulfuric acid by microbes. So soil transformation can occur due to ionic exchange, either through cation or anion exchange. In acidic soils, you have the soil where you have hydrogen ions or protons that can be replaced, that can replace all other cations. Protons can be replaced by low affinity cations like calcium also. But if calcium is present in higher concentration, or protons are in short supply. So we have a condition here that if we have a soil, this is the clay part of the soil, clay myceli, where you have the hydrogen on its ends, which are because of the aluminum negative charges on the clay, aluminum silicates on the soil, which has the negative charges and with which are attached the protons. So the soil becomes acidic. And on the addition of the lime, the calcium, which is low affinity, but once it is present in more concentration, it can replace the protons here. As a result, there is more calcium availability, it will become less acidic soils. So affinity of various cations to colloidal clay or humus may occur temporarily in ascending or descending order. It is called as Hoop-Mister series, or it is called as lyotropic series. Lyo means dissolve and tropic means change. So once we have a situation that soil can be an acidic, or at the same time it can be an less acidic, depends upon the availability of the other cations. So it means concentration dependent physical effects in solution or ion specific behavior in aqueous solution. Hoop-Mister series makes it understand that if you have a proton which is greater than hydrogen concentration, hydrogen ion concentration is greater than calcium ion concentration, the hydrogen will prevail there. The soil will be acidic. If calcium is greater than the magnesium, so it will be less acidic soil. So magnesium can be greater than potassium, or this series is called as the Hoop-Mister series, and it means the concentration dependent physical effects in a solution or ion specific behavior, how ion will behave in an aqueous solution. So that means you can make an acidic soils into less acidic soils and less acidic soils into an acidic soils. What is the mechanism of mineral absorption? Plants absorb mineral ions from soil through root system. Plants have a massive root system and it acts as an apparatus for absorption of mineral ions from the soil. As we have already said, plants act as the miners on the earth's crust because they absorb minerals through their root system. Ion absorption takes place through thin-walled cells of, we have a root apex, zone of elongation, and root hair zone. In a root, you find a root apex, a zone of elongation, and root hair zone. Ion is absorbed by root apex and zone of elongation, create an osmotic potential in roots. Ion is absorbed by root hairs or translocated to the shoot. because there is to be an lateral conduction and the vertical conduction. So the ions absorbed by these cells will create an osmotic potential in the roots. And if it is directly taken by the root hairs, that will go to the shoot system, translocated to the shoot. Movement of mineral ions along the water occurs through the epidermis, cortex, endodermis, and xylem. As we have seen during absorption of water by plants, it can be either through apoplastic movement through cell wall, or it can be symplastic movement through cytoplasm. So apoplastic movement through cell wall, it's a rapid movement and it is a passive transport. Whereas symplastic movement through cytoplasm, it is a slow movement, an active transport involving metabolic energy in the form of ATP. So roots utilize metabolic energy for this symplastic movement of mineral ions. So you can see the simplified diagram. This is the root hairs. This is the exoderm cell, and the cortex cell, endodermis cell, and this is the stele. It will be having the xylem cells here. There will be Casparian strips here. So apoplastic pathway will be up to this, and then there can be a transmembrane pathway which will be all along, which will take place through the cell walls, through the plasma membranes, or there can be a symplastic pathway which will occur through the plasmodesmata and will cross from one cell to the other cell through the plasmodesmata. So we have in plants 3 modes of absorption: transmembrane pathway, symplastic pathway, or there can be an apoplastic pathway. Mechanism of mineral absorption, as will be focusing on the passive mineral absorption. That means mineral ions are absorbed into living cells through diffusion. No ATP is used. Active mineral absorption, which we will discuss in the next video, is absorption of the mineral ions against concentration gradient, uses metabolic energy. So this is as per the gradient, that's from higher to the lower, and this will be against the concentration gradient. Passive mineral absorption, it involves the movement of mineral ions into living cells by diffusion. It occurs along the concentration gradient, and it fails after the establishment of an equilibrium. Obviously, when it is as per the concentration gradient, when there is an equilibrium, establishment of the equilibrium, that time there will be no movement, no diffusion, no ATP is used by plant root cells. Diffusion of ions across root cells is only possible when ion concentration is higher in the external medium. Then it will be from higher concentration to the lower concentration as per the gradient. It can be maintained indefinitely by 2 ways: by immobilization of solute inside the cytoplasm or cell sap, by maintaining an electropotential difference over the membrane. So passive absorption is one which is as per along the concentration gradient, does not use the ATP, and plants, it is only possible when internal concentration and external concentration gradient is maintained. Various modes of passive mineral absorption in plants are: there can be a simple diffusion. It can occur by 2 methods. It can cross lipid matrix diffusion. As here, non-electrolyte like ethyl alcohol, methanol, and lipid-soluble ions like oxygen, carbon dioxide, ammonia, etc. They pass through lipid bilayer. It is a simple diffusion as per the gradient. Second is aquaporine diffusion. As we have already gone through the aquaporine, which are present in the root cells, which are called as the water pores also. It occurs in root cell membranes. Diffusion diffuses water-soluble gases, oxygen and carbon dioxide, small ions through water pores or aquaporin. So whenever there is a water moving en masse as a mass flow due to transpiration pull, plant roots absorb water due to transpirational pull, which is in the aerial foliage. As a result, aquaporins allow the passive movement of ions along the mass flow. When there is a mass flow of water, along with there can be the passive movements of ions also. That is called as the aquaporin diffusion. You can see with the simplified diagram, this is the plasma membrane. These are the lipid bilayers. Here, a non-polar oxygen, carbon dioxide, ammonia can cross the lipid bilayer. It is called as a simple diffusion. Or you have the aquaporins here, where outside ions can cross inside, which will be as per the mass flow along with the water. Or there can be a facilitated diffusion that we will see that is taking place either through channel specific, or there are certain transmembrane proteins which are called as the permeases. They allow the movement from outside to the inside. They are specific in nature. And you will see the discussion part of it in the next slide. Or you have a simple diffusion here. This is the plasma membrane, lipid bilayer and protein bilayer. And with passage of time, you will see there's an extracellular fluid. It's crossing the plasma membrane, lipid bilayer. And with passage of time, you will see the equilibrium maintained there. So it's a simple diffusion across the plasma membrane. Facilitated diffusion is mediated by certain transmembrane channels, protein carriers like permeases. Membrane channels are transmembrane regulated pores formed by proteins. They are specific in nature due to unique characteristics like pore size, electric charge. They carry ions at the rate of 10⁸ ions per second. Example, they can carry potassium, magnesium, nitrite, chloride, and phosphates. So diffusion facilitated by means of certain transmembrane channels or by means of protein carriers is also possible in the plants. Protein carriers are permeases. They are structurally like carriers which function like enzymes. So soon after contact with solute particles outside, they undergo 180-degree conformational changes. Ions are released on the inner side of the membrane, just like enzyme and substrate complex. When substrate comes in contact with the enzyme, there's a conformational change in the enzyme. So it rotates and carries it to the inside. Enzyme reverts to their original position for next translocation after releasing it to the inner side. It's a slow transport carrying 100 ions per second. It is 10⁶ times slower than the channel transport. So that means channel transport is quicker than the protein carrier transports. Here with a simplified diagram, you will see this is basically here a protein channel. These are the lipid bilayer, protein bilayer, and this is the extracellular fluid. These are the ions which are coming through it and going inside. Facilitated diffusion of ions across the ion specific channel. These are specific ion channels. And there can be, this is another specified diagram for the carrier proteins. So channels and proteins, there's a difference. So proteins are protein carriers. Once it comes in contact with it, there's a conformational change in it and it is rotating and pushing it from. So this is outside, and then once the protein comes into contact, there's a change and it is carried from outside to the inside. So facilitated diffusion takes place by means of these protein carriers which undergo a conformational change. If it is this way, it will turn this way. So it will release the ions inside, carrying the ions from the outside to the inside. So this is called as the facilitated diffusion. So mass flow, it involves absorption of some mineral ions by plant roots during heavy transpiration, as I have already mentioned in aerial foliage. It occurs along mass flow of water through aquaporins. transpirational pull acts as a driving force for this. So roots acts as passive apparatus for mineral absorption. Mass flow of water may carry some salt ions with it from the root area. Water-soluble gases, oxygen and carbon dioxide, and some small ions are absorbed. Leads to depletion of salts in root, resulting in a steep chemical gradient. Thus favors further absorption of minerals from the soil. Direct correlation exists between salt uptake and rate of transpiration. So there is a transpiration which is creating a transpirational pull, and you will see there is also the salt uptake. That means the minerals are also taken. That means with mass flow, there is also an uptake of the minerals with the rate of transpiration. So transpiration pull is creating a mass flow of water. With this, the minerals are also taken inside. There's an ion exchange taking place. It refers to exchange of ions with equal charge between the root cells and the external solution. So there can be a cations by cation between roots and the soil. Roots exchange protons for cations and anions for anions. Ionic exchange has been experimentally proved in barley roots. See, a barley root was fed with a potassium bromide solution having radioactive K. So a barley root was taken and it was fed with a potassium bromide solution, which was having potassium was having a radioactive isotope. After some time, the root was excised and was placed in a distilled water. There were no traces of radioactive potassium in the medium. But when same root tip was next immersed in a solution of potassium bromide, radioactive potassium ions were noticed in the medium. The concentration of potassium ion remained constant in the external solution. It indicates that equal number of non-radioactive potassium ions have entered into root in exchange of radioactive ion leaked into the medium. That means there is an ion exchange taking place. It also indicates that positively charged cation potassium is exchanged with similar cation potassium, though it is radioactivity with a radioactive isotope. That's why in a medium, when it was fed with potassium bromide, and it was found that radioactivity is noticed in the medium. Ions, both cation and anions, are readily available on the surface of soil membranes of roots. So it will be explained with the help of the next slide, where you will see that there is an ion exchange taking place. So ions are being exchanged here. Ion exchange mechanism is there. Ion exchange mechanism can be explained by two theories. Contact exchange hypothesis, which was proposed by Jenny and Overstreet in 1939 and 51. As per this hypothesis, a direct exchange of ion between the root cells, because root is present in the soil, minerals are present in the soil, there has to be some exchange, and either the ions are with the colloidal soil particles, or that will be in the form of a solution there. So whenever the root comes in contact with the colloidal with these ions, both overlap each other, because ions are not static. They are always in oscillation. They are always oscillating around their absorption surface. So roots and clay particles are in close contact. Whenever they come in close contact, and when their oscillation volume of the cations of roots, suppose protons, and that of clay particle overlap, ion exchange takes place. So as the literal meaning is, that is ion exchange. The ions are exchanged. How they are exchanged? As per this hypothesis, simply explained, if this is the epibelma, this is a root hair, extension of the epibelma, and this is a clay myceli which has a potassium on it, and this potassium here is oscillating with the clay myceli. This is oscillating here. So these ions are oscillating. Also, the root here has a protons which are oscillating on their root surface. Whenever there is a close contact between the clay myceli and this root, and that contact is here, and their oscillation area overlaps. Whenever there is an overlapping of an oscillation area, the element which was present here is taken to the clay, and from the clay the potassium is taken into the roots. That is why it is called as the ion exchange, and it's explained by means of a contact exchange hypothesis. That is, when contact is established with the root, with the clay, there can be an exchange of ions, that is cation can be exchanged with the cation. So this is called as the contact exchange hypothesis. Second is explained with the help of carbonic acid hypothesis. During respiration, roots release carbon dioxide into soil. Carbon dioxide combined with water forms carbonic acid, and carbonic acid dissociates into proton and bicarbonate. And proton is exchanged with cation held by the clay particle. Cation with bicarbonate are released in the vicinity of the root surface. Due to availability of higher concentration of potassium bicarbonate, root cells absorb potassium bicarbonate. So with the help of this carbonic acid hypothesis, which is explained with this simple diagram, this is again the root epipelmal layer, this is the root here, the clay soil, the clay colloidal clay here is having potassium on it, which is always in an oscillation, but roots, they undergo respiration due to their metabolic activities, and they release carbon dioxide. That carbon dioxide combines with the water in soil, forms carbonic acid, and carbonic acid is dissociated into bicarbonate and protons. Now, this proton comes in contact with the soil clay, which is oscillating. Here with the oscillation, there will be the replacement of this with the potassium, and potassium will combine with the bicarbonate. Higher concentration of the potassium bicarbonate in the vicinity of the roots, it will be absorbed. So there will be an ion exchange. The potassium will be taken and in exchange of the proton which is in the colloidal clay. This is called as the carbonic acid hypothesis, and it also helps us in an exchange. One more thing which explains the passive absorption of minerals in plants, that's called the Donnan equilibrium. Donnan equilibrium was named after British chemist Frederick George Donnan, 1870 to 1956, who proved its existence in biological cells. That is, Donnan equilibrium is existing in biological cells. Before this, Willard Gibbs had already predicted the same effect. That's why it's also called as a Gibbs-Donnan equilibrium. It depicts the behavior of charged particles near a semi-permeable membrane that sometimes fail to distribute evenly across the two sides of the membrane, or it describes the equilibrium that exists between two solutions separated by selectively permeable membrane. It explains the passive accumulation of ions inside the cell. As per this theory, there are pre-existing non-diffusible ions inside the cell, which are named as, non-diffusible ions, which are also named as fixed ions. They include charged carboxyl, amino groups of proteins, or charged groups of other macromolecules in the cell. The cell inside has certain fixed ions, fixed molecules, which are non-diffusible. They will not be taken across the membrane because of the selective permeability of the membrane. So there will be always certain ions inside. So two forces govern the movement of charged ions. One is the concentration gradient, that means the actual number of ions. Second is the electrical gradient, that is the net charge across the membrane. So Donnan equilibrium explains it. We explain it here. Suppose there are same number of charged ions on both the sides of a membrane and there are non-diffusible anions on the inner side. At equilibrium, the concentration gradient of ions is balanced by electrical gradient, so the net driving force becomes zero. The concentration of ions is different on each side. Inside, potassium is more and chloride is less. On the outside, it is opposite. In the presence of non-diffusible ions, which are also called the fixed ions, which can be proteins, the diffusible ions distribute themselves so that at equilibrium the concentration ratios are equal. The process will continue till the multiple of free cations and anions become equal on both the sides. We can explain it, the Donnan equilibrium. Suppose we have a cell here. This is the cell inside. This is the outside. Outside we have charged ions. And this is separated by means of a semi-permeable membrane. Inside the charges, potassium is 5, chloride is 5, proteins are 6. So this is a protein, these are the negative charges here. Across you will find the similar charges which will be positively charged, and outside charged ions are potassium 5 and chloride 5. This is a hypothetical example with which we can explain the Donnan equilibrium in the best way. So we have an inside, we have an outside. Inside situation is that chloride, potassium is 5, chloride is 5, proteins are 6. So what will be the net charge here? This is balanced here, so net charge is 6. Here outside we have potassium 5, chloride 5, so net charge is 0. Now there will be an electrical gradient. As per electrical gradient, what will happen? One potassium molecule can cross here, and that leads to the formation of potassium as 6, chloride 5. Proteins are 6 there, but what will be the net charge on the two sides now? Since one positive charge has entered here, so there will be net charge as 5 now. On this side, there will be an imbalance in the charge, that will be 1 now. Net charge instead of 0 will be 1 here. What will be the charge difference? That will be 1 here, 5 here, that will be 4. And it will help here since there will be a concentration gradient now. Concentration gradient is favorable towards this side because there are more 6 potassium, here are 4 potassium. So the concentration gradient will be 2. A situation can again change. We have here a potassium 6, chloride 4, proteins can be 6 here, but what is the net charge here? Net charge is 4, and when the net charge is 4, there is a possibility that chloride ion can cross from this side to this side as per the electrical gradient. Once chloride has gone here, it has again caused the imbalance in the charge. There will be one more net charge here, it will be this. So as a result, there will be 2 net charges here and 4 net charges will be on this side. So this electrical gradient, which will be 2 here, but at the same time, it will favor the concentration gradient. We have here 6 and here we have 4 now. So there will be a concentration gradient of 2. So concentration gradient and the net charge will become the equal. So that means there can be an establishment of an equilibrium. At equilibrium, what will happen? There will be 6 chloride and there can be 6 potassium. Chloride will be 4. Proteins will be 6 there. And here it will be potassium, chloride, 4 and 6. So that means an equilibrium can be achieved here. But at the equilibrium, what will happen? There will be the net charges. It will be more on this side, less on this side. Let us explain it this way. A Donnan equilibrium is attained according to the following equation. You have a potassium outside, potassium inside, chloride inside, and potassium chloride outside. Or we have a now situation that is outside it is 4 and 6. That is 4 is potassium, 6 is chloride. Inside it is 6 and 4. So it will be net 24 by 24. However, number of the total ions are more on the side with non-diffusible ions. Because you have potassium chloride, you have proteins. Here there will be 6. 6 plus 4 is 10, but you have a 6 that is 4, that is 16. It is greater than 10. So on this situation, what will happen? There is a movement of ions, but no energy has been used here. Number of osmotically active particles is more on the inner side. When osmotically active particles is more on the inner side, because of these non-diffusible ions, it favors endosmosis. Water will move from outside to inside. In a plant cell, swelling can be contained by rigid cell wall because there will be a turgor pressure. There can be a swelling, but since it has a cell wall, it will not result in the rupturing of the cell wall. But in case of the animal cells, swelling can be followed by rupturing. However, it has a defense mechanism there. It can be prevented as cell pumps out more ions than it brings in. That is, there is a pump which pumps out 3 sodium and brings 2 potassium. So at equilibrium, there remains some difference in charge across the membrane because of these non-fixed ions, because of these fixed ions which are non-diffusible ions within the cell. A potential is developed across the membrane. The potential generation is passive. See, cell has generated a potential across the membrane, but it is passive as no ATP is required and no ATP has been utilized. It is a passive distribution of ions which generates a potential difference. So Donnan equilibrium may or may not necessarily occur in plant cells, but observations on the surface potentials of plant roots reveal that ionic distribution across the exterior walls of outer cells of roots is in accordance with the laws of Donnan equilibrium. It serves as explanation for passive accumulation of ions against a concentration gradient in response to an electrochemical potential gradient. So the Donnan equilibrium amply explains that the passive accumulation of ions can take place though against a concentration gradient without utilization of energy, but by creating an electrochemical potential gradient. With this, we have seen that different methods, different mechanisms of passive absorption. In case of plants, we have seen the diffusion, simple diffusion. We have seen how facilitated diffusion is taking place in the plants, how ion exchange mechanism is operating in the plants, and how Donnan equilibrium also explains the passive mechanism of mineral absorption in the plants. I hope this video will help you to understand the mineral passive modes of mineral uptake by the plants. I think you have understood this, and till the next video is uploaded, that will be about the active absorption of the mineral ions. Till then, be safe. Thank you for watching this video.